we've known for ages, right, that cancer can run in families, that it's written somewhere in our DNA. But how, exactly? How does our genetic code actually dial our risk for cancer up or down? Well, for a long time, that's been a massive puzzle for scientists. Today, we're looking at a huge breakthrough, one that doesn't just add a new piece to that puzzle, it kind of flips the whole board over.
So let's just get right to it. The big question has always been this: how does our DNA really affect our cancer risk? I mean, we get our genes from our parents, sure, but we all know that can't be the whole story, can it?
And you know what? It's not. The answer, it turns out, isn't just in the genes themselves. Nope. The biggest clues, they were hiding in plain sight, in these huge, mysterious stretches of our DNA that for the longest time, scientists just called junk DNA. Yeah, and that is where things get really, really cool.
All right, so first, let's lay out the problem that's been bugging scientists for years. We'll call it the cancer gene puzzle.
Okay, so here's the deal. For years, scientists have been running these huge genetic studies. They're called GWAS, basically scanning the DNA of millions of people to find tiny variations linked to diseases like cancer. And they were successful. They found over 1,000 genetic markers, little red flags in our DNA that are tied to cancer. But, and this is a huge but, most of them weren't actually in the genes. They were in that non-coding DNA we just talked about. So you can imagine the frustration, right? They had a map with all these X marks the spot locations, but no idea what treasure was buried there or how it was connected to the disease. It was a massive roadblock.
So to get past that roadblock, researchers had to think differently. They had to look beyond the genes themselves to a kind of biological control system, a system we can think of as a gene dimmer.
Now, the proper scientific name for this is DNA methylation. All it is, really, is a natural process where the body sticks these little chemical tags onto the DNA. Think of them like little sticky notes. The crucial thing here is that they don't change your actual genetic code. They don't rewrite the instructions. They just, well, they control how loud or quiet those instructions are shouted.
This light bulb and dimmer switch analogy is just perfect for it. Your gene, that's the light bulb. It's there. It has the potential to shine. But the DNA methylation, that's the dimmer switch on the wall. It can turn the gene all the way up so it's super bright, turn it down so it's just a faint glow, or switch it off completely. It doesn't change the bulb, it just controls it. And this control layer, this was the key they needed to crack the whole thing wide open.
Okay, so the researchers have this dimmer switch idea. But how do you actually connect that to someone's cancer risk? Well, they came up with a really clever 4-step plan to do just that.
First, they took a bunch of normal, healthy tissue samples to see how the DNA and these dimmer switches usually behave together. You got to learn the rules before you can find what's breaking them, right? Next, they used that info to build these really smart computer models that could look at someone's DNA sequence and predict where all the dimmer switches were set. And then the grand finale. They took these models and applied them to huge genetic databases from over 1 million people looking for patterns. They were basically asking, can our predicted dimmer settings explain who gets cancer and who doesn't?
And they didn't just look at one type of cancer. They went big, focusing on 7 major ones: breast, colorectal, lung, ovarian, prostate, renal, and testicular cancer.
So what do they find? Well, this is the aha moment. The results were, honestly, they were stunning. They found thousands of new clues.
Get this, they uncovered 4,248. Let that sink in. 4,248 potential new links. That's thousands of places where one of these dimmer switches seems to be directly tied to cancer risk. I mean, that's not just a small step forward. That's a giant leap.
But what's even more mind-blowing is how specific these links were. Look at this. Over 95% of them were unique to just one type of cancer. That means the dimmer switch setting that affects lung cancer risk is probably totally different from the one that affects breast cancer risk. It's not a master switch. It's an incredibly tailored system unique to each part of the body.
And if that wasn't enough, they also found 55 brand new hotspots for cancer risk, places on our genetic map that the old methods, the ones just looking at genes, had missed completely. It just goes to show you, you have to look at the whole system, the dimmer switches included.
Okay, so numbers like 4,055 are impressive, for sure. But what does this actually look like? How does a single dimmer switch actually connect to cancer? Let's connect the dots with a real example.
Let's zoom in on a specific chain of events they found for breast cancer.
Okay, so check this out. They found a specific spot on the DNA where more dimming, more methylation was happening. Step 1. This extra dimming caused a nearby gene to get much quieter, lowering its activity. Step 2. And step 3? That lower gene activity was directly linked to a higher risk of breast cancer. See? It's a clear causal chain. The dimmer switch setting changes, which changes the gene's behavior, which in turn changes the person's risk for the disease. It's beautiful.
So now that we've seen how it works on a small scale, let's zoom back out. What does all of this mean for the big picture? What's the future of how we think about cancer risk?
So, why does this matter so much? Well, for one, it gives us a much richer, more detailed map of how cancer is wired genetically. In fact, it found risk signals in 143 places where the old methods found nothing. Zilch! This could be huge for developing new tests, new biomarkers to spot cancer risk way earlier and more accurately. But at its core, it just gives us a much deeper look into the incredible dance between the genes we inherit and the way our environment and our bodies actually decide to use those genes.
And look, it's not just me saying this. The researchers who did the work said it themselves, and I quote, "These findings substantially advance our understanding of the interplay between genetics, epigenetics, and gene expression in cancer etiology." In other words, they know they've hit on something big.
So all of this leaves us with one pretty profound thought to chew on. For decades, we've been obsessed with reading the letters of our DNA, the A's, T's, C's, and G's. But maybe the real secret to cancer isn't just in the code itself. Maybe it's in how that code is being used. It raises a huge question for the future. Could we one day predict our cancer risk not just from the blueprint of our DNA, but from watching how those dimmer switches are being flicked on and off every single day?